So, what you see before you right now is going to be your new best friend. It's your inquiry explanation tool. We're using this for the results of all of our labs, and I'm here to walk you through it. First of all, here at the top, you'll notice a spot for your research question. This is the question you're asking in the study. Now, for instance, in the study that we're doing about macromolecules, your question would be which what sorts of macromolecules are in a Chick-fil-A meal? So that's your research question. What sort of macromolecules are in a Chick-fil-A meal? So that moves us to the design variables. You have your independent variable and your dependent variable, or your IV and your DV. Remember, the IV is the one that you choose, what I do as the researcher. So the independent variable is which food items I analyze from the meal using chemical indicators. So it's which food items I choose from the meal to analyze. The dependent variable is the reaction of the indicator chemicals showing what those things are. Showing what those things are. Is a reaction of the indicator chemicals, which then will show us what macromolecules are present. So the independent variable I pick, which food I analyze, determines the dependent variable, which nutrients are actually in it, and therefore which chemical indicators get triggered. Now, in terms of new vocabulary, you'll want to stick in here any words that are relatively new to you. There are probably at least two or three that we use over the course of the lab that will be new. I want you to write down the word and give a short definition. Next, we have the experimental steps. This is the design or the procedure for the experiment. So, for instance, step 1 would be choose which food you wish to test. Step 2 would be place the shredded samples in test tubes with water and 20 drops of the indicator chemical, or something along those lines. But the experimental design steps is the step-by-step procedure of the actual experiment. In it, you would say how many grams of the sample you put into the test tube. You would say how many drops of the indicator chemical, how much water you used. All of those things go into the experimental design steps, including watch the indicator or stir up what's in the test tube until the end and wait for the indicator to change color, indicating what you actually have in there. So the design steps, you start with usually something having to do with the independent variable or the choices in the independent variable, and they go on through to, at the bottom, the last one is almost always record your results. Record your results somehow. Now, as we look at the experimental design steps, the constants, it's not that you only hold these particular things constant. In science, you always want to make each experiment only have a difference of the variable you wanted to change. You want the only difference to be the independent variable, that all other possible variables are controlled. But in most experiments, there are a few issues that you really have to keep a watch on. So, for instance, in this one, it might be the volume of the food samples, that you don't want to put half of a chicken sandwich ground up into one test tube and like a little teeny tiny grain of bun into another one, or a tiny piece of cheese in one spot and a great large amount of Chick-fil-A sauce in another test tube, but that you want to make sure that the volume of the samples are all the same. Other constants that you can talk about would be constants like temperature, or always using indicators from the same container or something along those lines. Science concepts basically have to do about things that you read. These are the big scientific ideas the experiment is connected to, such as in this one, it might be the idea that different macromolecules are shaped differently and do different things in the body, and so they react to chemicals in different ways.